This video will begin our discussion on conservation genetics, focusing on its correlation with genetic drift. To be able to understand conservation genetics, we need to understand a little bit about conservation as well. So this is thinking about a scientific field in which we are looking at understanding and potentially counteracting the rapid loss of biodiversity. Now, when we think about biodiversity, we're commonly thinking about it from three different standpoints. We're thinking about it from an ecosystem standpoint, species, as well as a genetic standpoint, thinking about diversity between those. So it should make sense that as we think about this correlation between conservation genetics and drift, that they go hand in hand. Because when we think about the overall genetic diversity, we're studying within populations and between populations. So specifically thinking about conservation genetics, this is a subset of population biology, where we're looking at how to apply genetic information to preserve species that are capable of coping with ecological changes. More specifically, if we think about this, we want to think about the genetic management of populations based on their size, particularly how populations and their genetic diversity differ depending on the overall size of individuals that are available in that particular area at a given time. So when we think about this, we want to think about how genetic drift ties into this. Well, first and foremost, we need to think about the idea of the effect of a finite population size on gene frequencies. This is really important. And when we think about finite, remember, we're thinking about an actual numeric value that we're giving into place here. So when we think about the idea of genetic drift, remember this differs from the other two mechanisms that lead to evolution, like natural selection and gene flow, and that there's a true random aspect to drift, because we're looking at the random change in gene frequencies from one generation to another. And these changes typically correspond with large fluctuations in population size. And when we think about these large population fluctuations, think about the things that may lead to them. We've talked about catastrophic events leading to things like a bottleneck effect. We've also talked about the idea of looking at founder effect, where a small subsection of a population will migrate away and recolonize. The concept of thinking about the mainland species in relation to our island species. As we think about this, I want you to think about how overall population size corresponds with the amount of alleles in a gene pool and how those could potentially change over time. So if we think about the general concept of thinking about allele availability in a population based on their size, we can look at the simulations that have been provided for us. So thinking about a finite population, we can think about the fact that adults in a particular generation will pass on a finite number of gametes and their corresponding alleles to produce offspring for that particular generation. Those allele frequencies in that gene pool will generally deviate from the population's initial frequency and change over time due to chance events like genetic drift. So when we think about this, it should be no secret. Parents will pass on their genetic information to their offspring, and they do it by passing on parts of that information in their gametes. Diploid organisms will have haploid gametes. So half the genetic information will be passed on during the idea of reproduction, sexual reproduction, that is. So if we think about two haploid organisms, male and female respectively, a sperm and an egg will come together, each providing half of the genetic information, some from dad and some from mom. When that comes together and fertilizes, we result in a diploid organism, which has different genetic information than each one of the parents individually, as it is now a combination of that genetic information. When we're thinking about the combination, let's think about nuclear DNA as an example. So when we are looking at the simulations here, I want you to think about potential alleles in a population and how they will change, number one, over time, and how that frequency will not only change over time, but will it show the same overall trends depending on the overall population size. Now, in these examples, we're going to focus on effective population size. So we're physically focusing on the individuals in the population that are actively breeding to produce offspring generation after generation, right? We can think about the idea here that at a particular locus of a gene, multiple neutral alleles, remember neutral, that's the idea here that there's no selective pressures, and if they are, they're identical on those particular alleles, so there's no preference. Genetic drift will lead to fixation of one of those alleles in the population and loss of the other alleles. I want you to think about how that corresponds with heterozygosity over time. So if we look at these particular simulations, let's start out simply by looking at the largest population. So traditionally, we would move from left to right. In this example, we're going to focus on moving from right to left. So in our initial graph, right, these will be the same on all of the respective graphs. When we look at the axes, we will look at the y-axis that is corresponding with the overall allele frequencies, the x-axis looking at the generations over time, and then each one of these individual lines corresponding with a particular different allele. So the idea here is that when we look at our population of 2,000 breeding individuals, we're looking at 4,000 different alleles, 2 alleles per individual for a particular gene of interest. One of the things I want you to notice, and this is roughly the same on each one of the graphs, regardless of the population, is that initially we're going to make the assumption that all of the alleles start with roughly the same frequency, roughly about 0.5 across the board for each one of them. I want you to note, moving away from those first couple generations, how does that frequency change in each one of those colored lines representing the different alleles? If we look at our population of 2,000 individuals, notice the allele frequencies are relatively stable, suggesting here that they remain roughly the same from generation to generation over the course of the 50 generations that we're seeing. Very little variation off the 0.5%. After all, even after 50 generations, all alleles are still present in a population and all have roughly somewhere between 0.4 and 0.6 as far as their frequency. If we move over to our middle, where we are looking at a reduction in population from 2,000 to 200. Notice what we see. The trends here on this particular graph are drastically different than what we observed before. Again, starting roughly at about 0.5% frequency for all of the alleles, notice that over time, those frequencies become more sporadic and spread out. The idea here is that though nothing has reached fixation and nothing has lost, we're seeing a drastic change in the frequency of some alleles in comparison to the other. So seeing this randomization over time occurring, where some of them appear to have higher frequencies than others. And what does that mean? Well, let's take a look and see if we can reduce the population yet again and see what kind of effect that we see. If we look at our graph furthest to the left, we are observing our population of 20 breeding individuals. In this case, once again, note that our frequencies are roughly all starting around the 0.5 to keep things consistent. Look at the changes over time. Just like in our population of 200, changes get more drastic the further we go through generations. But changes in our smaller population appear to be immediate in comparison to our larger populations. As we can see, there's a drastic shift initially between the first 10 generations between the allele frequencies, where some are higher than others. We will even note if we move down further into between 15 and 20 generations, we see some very drastic changes. We see some of those allele frequencies are listed at being 1, suggesting that those particular alleles have reached fixation, a change in the gene pool where only one single allele for a gene is remaining. That's something that's very interesting, because if we look below, we can also see that in the opposite end of the spectra, we have some of those particular alleles that are being phased out and that are being lost over time. So there seems to be a strong correlation in the rate of fixation versus loss with regards to population size. The smaller a population is, the more drastic this effect has on that population. And how does that correspond with heterozygosity? Well, one may think about the correspondent with the type of alleles that you're actually observing in this particular diagram. One of the things that's important to remember, in this particular diagram, we are focusing on the frequencies within a population. And in here, we can see that the change in population size, particularly the reduction from 2,000 individuals to 200 and to 20, simulating the effect of genetic drift between each of these populations, shows the fact that drift does decrease genetic variation over time, and that the smaller a population is, the more drastic or faster that particular overall effect is. One may ask, would this effect be the same if we are looking at the relationship between populations? Well, if we are looking at between populations, even though an individual population is seeing a decrease in genetic variation, it would suggest potentially that drift would result in an increase between populations, meaning they would be becoming more different from one another over time, hence potentially increasing overall genetic diversity if those two populations were able to breed with one another. Now, as we think about this idea of population size and randomization and the correlation between changes in allele frequencies, let's look at an actual example. Now, the example that we're going to be looking at is a famous study looking at Drosophila, fruit flies, a model organism that was conducted by Peter Bury in the 1950s. In this particular example, Bury was looking at breeding populations of fruit flies over several generations and making observations based on phenotype and then their corresponding respective genotypes. So as we think about this particular study, he was looking at how over 100 populations of Drosophila evolved over time. We're going to focus on 19 of those generations for this particular example. So in this study, what Bury observed, focusing on initial phenotypes, looking at eye color mutation. With the idea of eye color mutation, he observed 3 different mutations, solely focusing on color. They were white, pink, and red, respectively. Now, when we think about these particular ones, we observe the idea of 2 different alleles being observed. Down here on the bottom of our diagram, we can see one of these, the BW75. That was one of the alleles present. The other one was just straight BW with no 75. So in each one of these phenotypes, it was observed that those individuals with the white eye color had BW, a homozygous situation with regards to their phenotype. If we looked at the red variety, the other extreme, this showed us the example of BW75 for that example, another homozygous scenario. This resulted in our intermediate eye color of the pink being our heterozygous situation. In this case, it should be noted that that intermediate eye color is actually a result of a variation of Mendelian genetics known as incomplete dominance, where we have a blending of the 2 alleles that are being passed on from the parent. This should make sense if we think about one allele leading to the phenotype of a white eye and one allele leading to the phenotype of a red eye. The combination of those colors alone, when you mix white and red together, if they are mixed equally, will result in a pink coloration, hence where we're getting the pink eye color for this particular phenotype. Now, in this particular study, he focused on eye color mutation as this is one of the tried and true things to observe in Drosophila, particularly when we're looking at the correlation with genetics, because there's a strong relationship between them and different alleles. In this particular experiment, he observed 16 individuals over the course of 19 different generations. In each one of the generations, Bury randomly selected 8 males and 8 females to observe those phenotypes. He was initially suggested that in the starting population, there was a 50% frequency of each one of the eye colors. Thus, it was suggested that his initial observations resulted in 5 white, 6 pink, and 5 red-eyed individuals. When he made this observation, he returned those 16 individuals in total back into the population. The idea here was that he allowed those individuals to freely breed with one another, and then he observed the results in the next generation. Now, one of the things to think about is every generation subsequential from our first generation resulted in the same randomization. 8 males and 8 females were collected, phenotypes were recorded, and those individuals were returned to the population. When we think about this, we are consistently studying a total or consensus population size of 16 individuals. The idea here was that we wanted to observe how things were changing when we are randomly selecting individuals, seeing if there was a change in the phenotype that would correspond with the allele frequencies over time. And that's what you're seeing here in this particular graphic, is you're looking at the distribution of gene frequencies in the observation, starting with the first generation, migrating all the way down to the 19th generation, showing you the total number of, in this case, focusing on one of those single alleles, the BW75. Now, in this case, remember, we have 16 individuals total. This is counting up. Each one of these numbers is representing each one of the potential 32 alleles that were present. See how the distribution has changed within that generation as we migrate through. Now, with this randomization, one thing that I think is very important to keep in mind is when Bury was selecting the 8 males and the 8 females of each generation, there was no way of knowing if those were the particular individuals that were actively breeding to produce the next generation of offspring. And what I mean by that is the N, the total population size, if you will, was noted at 16. But if we actually take a look at the breeding populations in each one of those generations, it was noted that on average, it was 19.2 individuals that were part of the effective population. So those contributing to the gene pool from one generation to another. That's very important to think about when we're looking at this study, as well as when we think about conservation efforts in the wild, to think about the overall total population number in comparison to those individuals that are of reproductive age that are actually reproducing over time. Because as we think about that, that may have a correlation to the overall frequency of genes available or alleles available in that particular population. One thing that was noted through the study is that there was a faster fixation of one allele in comparison to another, showing that one of the eye colors seemed to prevail over the other. In this study, one of the things that was observed as well is he observed a total of 28 fixations and 30 losses of one of the alleles over the 19 generations, which is very interesting to take a look at. And this may lead you to think about how that corresponds the fixation versus the loss rate with regards to changing heterozygosity. So as we think about Bury's study, we want to think about the change in heterozygosity with regards to population size. When we think about this particular concept, this graphic is a good representation of that. In this particular graphic, we can see on the y-axis, it shows us the fraction of initial heterozygosity seen in a particular population, corresponding with generations over time. We can also see that each one of the lines representing the data is giving us an overall total population size. We can see starting with 1,000 and then migrating down to 20. It should be very easy to look at this particular graph and draw some conclusions. As we think about this, first note that over time throughout generations, regardless of population size, we see changing frequency of heterozygosity over time. It's the rate of that change that differs depending on population size. So when we think about this, we can think about the correlation here with the smaller the population size, the faster the decline in heterozygosity. So the greater the potential for loss and fixation of alleles is occurring in smaller population size. That should make sense because think about the overall number of individuals and the number of available alleles in that population. They go hand in hand. So the less number of individuals, the less number of alleles to potentially pass on, particularly when we're thinking about traditional inheritance with regards to dominant versus recessive. Now, as we think about this, is this what was seen in Burr's study as he was collecting data looking at those particular fruit flies? In this graph, this shows you an example of some of the data later collected by Burr as he was looking at the changes of alleles with regards to eye color in his fruit flies. As we look at this, we want to look at the theoretical versus the empirical data. Again, this graph is representing the heterozygosity frequency in a particular population on our Y and the number of generations on our X. Notice the amount of generations is drastically different than what we observed before, but we are observing very similar trends. In this particular case, we will note that there are 3 lines, 2 gray and 1 red. Each one of the gray lines are representing the theoretical data over time. In this case, we can observe the idea here that the dotted line is representing our theoretical predicted loss or change in heterozygosity based on a population of 16, in comparison to our predicted loss of individuals with 9. As we look at this, we can see that observing the actual data, where we were observing, once again, a population of 16 individuals, 8 males and 8 females over time. We see that over time, in this particular population, after generations, we still see a decline in heterozygosity. Though it should be noted that this decline in heterozygosity corresponds with closer to our predicted population of N than it does to our predicted population of 16. I want you to think about why you think that is. But the moral of the story here is that the smaller populations are, the faster there will be a loss of heterozygosity throughout different generations. When we think about this overall loss of heterozygosity, one thing that may come into play is thinking about the commonality of the alleles. Typically, when we're thinking about alleles in a population, we think about the idea of dominant versus recessive. And then we typically correlate with the idea of rare alleles versus common alleles, and so on and so forth. So one of the things you should also think about when looking at data trends such as this is you want to think about whether or not the rareness of an allele actually makes a difference overall on the frequency of its availability in a population. So as we think about this, you should see that there are similar trends to what we are looking at with population size and heterozygosity in correlation with the rareness of alleles. So if we look at our empirical data that's provided here, looking at the number of individuals in a population as it declines, we can see that there is a direct correlation in the loss of rare alleles with regards to that population. The larger the population is, the more alleles that are present after a single generation in comparison to others, which is very important to think about. Now, as we observe this, it's also important when we're thinking about the rareness of alleles. Remember, when we think about dominant versus recessive, recessive does not always correspond with the rarer allele between the two in the population. After all, we can have instances where individuals that are homozygous recessive are considered the wild type, the more common in a population. And think about what the correlation between that is, this, and potential selective pressures, as they may be very important to think about later on when we think about overall conservation, when we're thinking about the ecological ramifications or aspects in correlation to the genetic information. Now, and lastly, as we come to the end of this video, Keep in mind the potential consequences that we discussed of genetic drift, the idea of the potential fixation of alleles, the effective chance that varies between populations, particularly based on size, the effect that typically we see stronger correlations with the loss of genetic diversity in smaller population size, and that directly corresponds with the idea of reducing variability in populations by reducing heterozygosity. As you continue to study the subject, if you're curious more about the particular study that we focused on as an example, I have posted an electronic version of the paper that corresponds with that study from the 1950s. I encourage you to read it over. It's available in the content section for this particular chapter's material on my courses.